Hello everyone, myself Anugraha Ganesh TG from 2nd MSc Physics, Hindustan College of Arts and Science, Coimbatore. Today we are going to discuss about the topic FESM and Edax. So let's see what is FESM. The expansion of the term FESM is Field Emission Scanning Electron Microscopy, which is a technique that uses a beam of electrons to image the surface of a sample. It's used to study the composition, topography and other properties of the materials. The other importance of this technique is a high energy beam of electrons scans the sample surface. The electrons interact with the sample's atoms to produce signals. These signals contain information about the sample surface properties. Like this, the working is taken place. And there are few advantages about this technique. That is, it produces clearer images with less distortion, can examine smaller contamination spots, can produce high quality images at low voltages, can image insulating materials without the need for conducting coating. The other applications of FESM, that is, it can be used to study the morphology of nanocrystalline powders and bulk material. It's used to study the elemental composition of materials. It is used to investigate the structural organization of biological systems. It is used to study the interaction of parasitic protozoa with host cells. And here given is the pictorial representation of field emission scanning electron microscopy. Next we are going to discuss about Edax. The technique Edax is the abbreviated form of energy dispersive X-ray analysis. It is also known as energy dispersive spectroscopy, which can be abbreviated as EDS or EDX. Here EDX or XED, sometimes it is termed as energy dispersive X-ray microanalysis or energy dispersive X-ray analysis. This is an investigative method which is widely employed in elemental or chemical characterization of objects. Here, the sample may be of the form like a solid thin film, solid powder, liquid sample or even a pellet. Here, a typical setup of Edax is similar with that of the SEM setup. More specifically, Edax is actually inbuilt within the SEM setup. The working of EDX is based on electron focusing, same as in the SEM. Likewise, we understood that it is similar with SEM. Electron beam excitation is employed in SEM and scanning transmission electron microscope, whereas the X-ray beam excitation is present in X-ray fluorescence spectrometers. Here a detector converts X-ray into voltage signals which feed a pulse processor to measure the signals and pass them to data analyzer for display and investigation. Here silicon detector cooled at cryogenic temperatures by liquid nitrogen is often employed. Additionally, some modern systems come with silicon drift detectors employing Peltier cooling systems. To produce characteristic X-ray from an object, it is bombarded by either a highly energetic beam of charged carriers like electrons or protons. And the atoms of the sample contain ground state electron in discrete energy level or electron shell bound to the nucleus. An electron from an inner shell may be excited by an incident beam. Therefore, removing it from its shell and generating a hole where electron was present before excitation. Here the hole can be occupied by an electron of a high energy shell. The difference in the energies between the higher and lower energy shell is emitted as X-ray. Quantitative measurement of energy and number of these X-rays can be done with energy dispersive analysis of X-rays. Since the energy of these X-rays is characteristic feature of energy differences between the two shells and atomic structure of discharging element, Edax can be employed to identify the elemental composition of an object. And here the instrument is composed of 4 components like the electron beam source, X-ray detector, pulse processor and analyzer as seen in the diagram. Let's see how the quantitative analysis is taken in Edax. As characteristics X-ray intensities are proportional to the particular element's concentration, therefore quantitative analysis can be performed. Concentration of the unknown element in an sample can be obtained by matching the intensities of the characteristics X-ray of standard sample with that of the unknown sample. However, X-ray generated in the sample may be either absorbed within the sample or excite the X-ray from other elements before being emitted in vacuum. The correction is required in this analysis method, for which the following requirement is prerequisite. Flat specimen surface. Electron probe should enter the sample in perpendicular direction. Distribution of the elements in the X-ray generation area should be uniform. There are always some appreciable errors in this analysis method because some specimens do not meet these requirements. So we came to know about FSM and Edax. Thank you.